We're kicking off Unit 5 with a slightly different topic that, while doesn't quite mesh in with everything we've been talking about up to this point, at least from the surface, is an extremely important and current topic in biology, and that has to do with biotechnology. So why do we care about biotechnology? DNA and the genetic code are found in every single living thing, and we can actually use that information and use biotechnology to produce things like medications, foods. We can use it to diagnose disease, to treat disease, and understand the things that are happening around us in the natural world at the molecular level. So biotechnology really is just taking what we know about the natural world and applying it in a way that's going to be useful for us. So biotechnology is a field where organisms, cells, molecules, something to do with living things, is modified to achieve some sort of practical benefit for us as humans. And there's a lot of different applications of biotech. We use it in things like agriculture, we use it in our health industry, and we use it in forensics. There's other things we can use it for, but these are some of the big ones. Now, if we remember the central dogma of biology, we remember that we have the instruction manual, DNA, we go through transcription to create our message carrier, mRNA, and then from there use the mRNA going through the process of translation to create a product, a protein. Right? So DNA to mRNA to protein. That is what we call the central dogma of biology. Now we can use molecular tools to help us identify things like important genes or to move genes around either within an organism, from one organism to another, or within an apparatus. It's actually surprisingly simple to do this too, to insert a new gene into something like bacteria. It only takes a warm water bath. This is something that is actually done at the intro level. So for example, when I was in college in my first biology majors class, we did this as one of our labs. Yep, an 18-year-old is able to do this as long as they're given some instructions. So the process is relatively simple, but the challenge comes from creating the DNA that you want to insert. So that's something that as an 18-year-old I did not do. We were given the foreign DNA in what we call a plasmid, and that was used to deliver the new DNA into the bacteria. Now, when we take DNA from one organism and put it into another, right, from one species to another, we call it transgenic. So the organism is now a transgenic organism, meaning it has DNA from two different sources. And those plasmids that I just previously mentioned are little circular pieces of DNA that we use as a, quote, vector to deliver new DNA. So think of it as just a way to deliver DNA from one organism to another. And this is what we did and we used when we transformed our bacterial cells with a new gene. We used plasmids that were made in a lab by someone else. So there's four main tools of biotechnology that we're going to kind of go over. We're going to talk about the main kind of purposes between each of those tools. One, we have to be able to cut DNA. So we have to be able to remove sections, and specifically remove sections of DNA from a donor species if there's a certain trait we're looking at. So we have to be able to cut DNA with little, basically molecular scissors to cut away all of the excess DNA so we just get that gene. We can amplify small samples of DNA to make more useful quantities. So let's say you are interested in a particular gene or a particular trait or section of DNA, but you have very, very little of it. We can go through a process to amplify that to make a much larger amount. We need to be able to insert DNA into cells like bacterial cells or viruses. And we need to be able to grow those colonies that have that donor DNA. So these are the basic four things that we're doing. Not all of these tools are used in every single application. Some will only use one or two, maybe three of these techniques. It's a mix and match depending on what you're trying to accomplish. So let's look at how we can do the first task, which is chop. And we do that through the use of what we call restriction enzymes. Restriction enzymes function like sort of molecular scissors. So if we look here, we have this gene of interest in this darker green aqua color. And we have a restriction enzyme that is going to target a particular base pair sequence of DNA. So it's going to go for this particular sequence. It's anywhere where it finds this exact sequence, it's going to cut. So in this case, we're looking at ATCGAT. So anywhere where this enzyme finds ATCGAT, it's going to cut it. And so what we do is we basically cut the hydrogen bonds between those bases and then cut the sugar phosphate backbone at the points where you see these triangles. And so what we create is this section of DNA that has a few open spaces on one end and open spaces on the other. Now what we'll do is we remove this DNA that we want to take, right, with the gene that we're looking for. And these two ends here will be moved together and will then rebind together so that we don't fully destroy the DNA we're looking at. It's still somewhat intact. We just removed the gene of interest. The amplify technique is through what we call polymerase chain reaction, or PCR for short. In PCR, what we're basically doing is forcing a DNA molecule to undergo replication over and over and over and over again. And so what we do is we have this segment of DNA we're looking for in a solution, and we heat that solution up. When we apply heat, it's going to force the two strands of DNA to separate so that we become double-stranded, from double-stranded into single-stranded. From there, now we've forced the two strands away from each other, we then bring in DNA polymerase with the proper primers and a bunch of free nucleotides, and we allow the enzyme to start building on these free DNA strands. While this is happening, we're cooling it back down. And so as we cool it down, then the DNA polymerase will start going through the replication process, adding and building a new strand onto the original strands. And so what you end up with that first original segment, after you go through these 3 steps, you end up with 2 strands. And then we will do this process of heating and cooling over and over and over again, giving you millions, if not billions of copies of the sequence you're looking for. So here we started with 1, we go through our PCR process, we end up with 2. We heat it again and cool it again, then each one splits into 2 again, so we have 4 total. Another round of heating and cooling gets us 8. So you see how it doubles every single time. So if we did another round, it'd be 16. A round after that, 32. A round after that, 64. And you can see how it's growing exponentially. The third technique is inserting DNA using plasmids. So we have some sort of gene we are interested in that we want to use. We find that in our source DNA, we use the restriction enzymes, see how we're using other techniques as well, to remove that gene of interest and then introduce it into a bacterial plasmid. So we cut the plasmid open after removing the gene and then insert the gene of interest in to that plasmid. So now we have a circular piece of DNA with the gene we're interested in. And then this is as simple as just heating up the bacteria we want this plasmid to enter into, put it back into a bacterial cell, and now it's going to use that gene. It's not going to know that that gene is foreign. It's just going to see it and go, oh, I'm supposed to use this, and start using that gene. So any DNA on the plasmid that makes it into the bacterium is going to start making the product that the gene makes. So the gene codes for a protein, and since we've now put that gene into the bacterial cell, that bacterial cell will now make the protein. And so that's something we can use, and we have been using to make useful products, especially in terms of health, in making certain medications like human insulin. The plasmid will also replicate alongside with the host DNA, so every daughter cell that the bacterial cell makes will also have that gene. So not only did you modify that one bacterial cell, but you've modified all of the bacterial cells that are made after that. And so we do that through the process of making clones through binary fission. And in that process, there are some concerns with producing genetically identical cells, organisms, and DNA molecules, which we'll get into in just a bit. There is a relatively new tool called CRISPR that has potential to really change things in a person's lifetime. This is something to keep our eyes on. CRISPR is a relatively new tool in that it did not really exist when I was in college. It was brand new when I was in school. So take that as you will. But it's kind of like a cut and paste, right? So we can remove a gene that is faulty, that maybe is causing disease, and then replace it with a gene that is functional. However, this process is still new. It's still being looked at. It's not foolproof. So as of 2018, it suggests that 96% of people cannot be CRISPR subjects due to the immune system recognizing the gene-edited cells and marking them for destruction. So this is, at least as of now, is not something that we can really use in people, but it is something that we use in other organisms. So what does CRISPR do? And CRISPR is really part of what we call the CRISPR-Cas9 system when it comes to gene editing. What we do is there's a gene sequence of interest that we want to work with, we want to remove. And so what we do is we have this RNA guide that goes along with this DNA sequence. We use a plasmid that has the RNA sequence and what we call the Cas9 sequence. When we use this, we have the Cas9 enzyme, which is like molecular scissors. It's going to cut wherever that RNA guide is at. So when that RNA guide meets with the DNA sequence of interest, we can cut there. Once we cut it, then we can insert the new DNA. So it's almost like just another version of those molecular scissors where we can cut the DNA and insert something new. But again, this is something that is still relatively new, and so we're still studying it and looking at the applications we can use it for safely. As of right now, it is not used in humans necessarily, at least not ethically.